Morning, everybody. Nice to see you all. Thanks for being here. I want to share a really, really important question with you all to start this conversation this morning. And that is, what's the difference between pattern recognition and pattern discovery? We're trying today to help you see that there is a gigantic world available through pattern discovery, and you'll see that through everything you see and hear on stage. But starting today and going forward, we're going to live in a world of human-computer action where we can say to the computer, bring me something I did not expect. Bring me something new, something dramatic. And when you bring it to me, maybe I won't know what it is, but it could change the world. That's where we are, where we stand together today. And that's why we built Pattern Computer. So I want to jump to the proof of this, and I'm not the guy to tell you that, but we thought it would be better if we had people come to talk to you who are world experts, they're well known, and on our team or not on our team, but they have their own reputations at stake here, and they've come to talk about all the stuff that we've done with them and for them. I'd like to begin by asking Ben Brown to come join me.
All right, so I've had the pleasure of working with these guys for the last several months now on the project I'm going to describe today. It's been a tremendous amount of fun, and I'm going to tell you about our experience working with Pattern Computer to use machines to understand biomedical systems. So my background, I'm a computational biologist. I work on complex systems, whether that's trying to understand how an ecosystem functions, trying to understand the gut microbiome, or trying to model how cells work together or how genotypes work together. Anything really complicated is sort of the domain of the kinds of problems we tackle. And even when you're looking at something as simple as a single organism, a single human, common throughout the human population are at least 6 million genetic variants that occur in 20,000 genes. And even if these variants had individual effects, so that they just operated one at a time regardless of what other genes were doing, that would be a big problem. But of course, genes don't work in isolation. When you look at the possible combinations across genetic backgrounds, across all of these genes, the space of potential interactions among variants is 10 to the 170th. To put that in perspective, there are 10 to the 83 atoms in the visible universe. Okay, so this is a number that can never be explored by brute force. Clever exploration strategies are needed to find the important aspects of genetic background, of genotype, that drive human phenotype. So if you want to do something like precision medicine, like use what we know about the genome to understand health outcomes, you've got a very serious problem on your hands. But of course, it's a problem we'd all really like to solve. We'd love to be able to associate individual genotypes with specific treatment recommendations, or even with unique drugs that promote the most beneficial outcomes possible. So one of the first things these guys did is they asked, can we create better prognostic panels? Okay. There are lots of prognostic panels. It's a huge industry. Lots of folks are making these things. There's the Blueprint, the PAM50. So these are breast cancer diagnostic panels. What you do in these things is you take a sample of tissue and you extract RNA and you analyze it for gene expression. And you ask yourself, is there a unique signature of a particular cancer subtype? You really care about subtype because it's associated with survivability. It's also associated with particular therapeutic directions, right? So for the major breast cancer types shown here on the 4 columns of this heat map, we've got luminal A, luminal B, HER2, and basal, right? These are different types of breast cancer. And down the y-axis on this guy, all these different blocks, these are different individual genes. Blue means low expression, red means high expression. And as you can see, when you look at the genes in this panel, you get very different patterns in these genes for the different subtypes. This is very cool, but of course, and some of these are in the Blueprint PAM50, or some of these are in Blueprint, some of these are in PAM50. Many of them are unique. So this has an 89% accuracy. One of the things we're working with them now is trying to validate this, because it looks like if you do repeat sampling on a single tumor, you maybe push accuracy to 99% or above. And that's a subject of interesting research. Another very interesting thing is you can look at...
You said 99%.
Yeah, I said 99%. Yeah, for repeat sampling, yeah, absolutely. So this is a representation, another representation of the heat map, another representation of the cube. And you've got our two people, A and B. They're both in the red region, so we both say they have a poor prognosis, right? A poor prognosis. But we have more than just a one or two-dimensional view of this. So if you just look at this two-dimensional slice, okay, they're in a poor prognosis. But if only their level of expression of BUB1, that checkpoint kinase, if only that thing were a little bit lower, you'd be groovy and you wouldn't be so worried. Now I want to animate this for you. Can we roll the animation? So when you dive into the third dimension, you see that the architecture is actually much, much different than it looks when you have just the two-dimensional picture, which is in turn much different than when you have just the one-dimensional picture, right? When you look at this third dimension, when you condition on the expression level of check 1, you see that one individual is quite a ways in this gene expression space from anything like a halfway decent prognosis, whereas the other is right on the boundary between poor and favorable prognoses. Now, every single one of these genes has a preclinical drug designed to hit it. What this gives you is a map from gene expression space to what you might want to do to move person A or person 1 or person 2 from the space they're in now to a space with improved prognosis.
So a couple of quick questions, kind of summarizing. It has seemed to us in getting to this point in working on breast cancer, this is all about breast cancer, that we've found a new way to understand cancer, by which I mean how to see it and how to actually manage it in terms of a risk portfolio, which you just described. So that we could say, for the first time ever, maybe we can look at a risk portfolio more intelligently, have a more deep understanding of the risks. And for any given patient, put that patient onto that chart you just showed in 3 dimensions and see how that patient could get out of there alive.
Yeah. That's exactly the hope.
So our next guest, you probably all have already heard of. He's very famous and a good friend. And Dr. Lee Hood.
And it again gets back to the data and the importance of, from data, creating correlations and or patterns, and from those patterns creating hypotheses, and then doing perturbations. And I'll say more and more N of 1 perturbations in humans are really going to be key in understanding disease complexity and in analyzing it. And the only point that I would make is, to do this, we do need the sophisticated computer science that we've talked about here.
So given where you are now in what you've just described, and the dependency upon big data and finding patterns and pattern discoveries in that data, how do you see working with us?
Well, I think the point I should have made that was not clear is we do have an enormous amount of data, but in the fascinating things I've showed you here, we're just barely beginning to scratch the surface in what we can do with it. And I think you raise another possibility, especially for being able to integrate information at different biological levels, to bring us back to a network understanding of the nature of wellness processes and of disease processes. So I think anything new and exciting, that's what we're talking about today in pattern formation, I welcome and will joyfully collaborate with.
Thank you very much. Thanks, Lee.
Okay.
Dr. Larry Smarr is another very, very, very famous guy. He's done a lot of different things in his life, ranging from general relativity to what he's doing now, which is running the Cal-IT2 Institute, which is probably the most amazing institute in the country. Larry, would you like to come out?
These are protein families, 10,000 protein families across the microbiome. And when you then do the analysis on those, you separate things out. So I gave this data set then to Pattern Computer. And over the last few months, I've worked with their technical team to say, if you had this data, what would you discover that I haven't seen? And the first thing they did is, by using the Pattern Computer, they were able to take this 10,000 variables per each of my 60 people and say, well, there's 39 out of those 10,000 that actually preserve most of the structure. That's That's an amazing reduction, almost 3 orders of magnitude. Then they were able to correlate those using the classic Spearman cross-correlation analysis. And you can see the 4 blocks down to the diagonal are at lower right, the healthy people, and then upward through the 3 disease states. You'll notice those 2 horizontal yellow bars where it seems like there's something different about them. And so when we go back and look now at a 3-dimensional version of that principal component analysis, that clustering diagram, you can see that on the left there, that the blue, the ulcerative colitis, completely separates away from the red ones, which are the colonic Crohn's, that's me, and then the ileal Crohn's, and then the healthy are all over to one side. But what is that group in the middle? In other words, that sort of seems like those are patients who might be in a region that's close to disease, just like we saw with Ben Brown, where those 2 people, one was close to being healthy and the other was quite deep into it. Well, that's the same thing you're seeing here. And so what the pattern computer people came back and said, Larry, we need to look at this yellow bridge set of samples, which didn't occur to me before to think about that. So we said, okay, let's take these 39 of the 10,000 KEGG protein families that the pattern computer identified as important and say which of those show a gradient across that yellow space. So these are which of those 39 protein families seem to have a directional change from disease to health. And here you just see one of them, and that's the gradient on the left. And you can just see it's a beautiful pattern that was completely unknown to me. And then on the left, you can see the colors are going essentially in the color of the rainbow from one side to the other, from the healthy side to the disease side. And here's 2 more. And again, you're finding these that show this gradient across there. So we've now selected 9, just 9 out of 10,000, so a 1,000-fold smaller number of variables to look at. And what we find is that these that were selected by the pattern computer were discovered are associated with oxidative phosphorylation, which is a disease, actually, if you don't have enough of those proteins, and is involved in a variety of the things that are associated with disease. So summing it up, basically, there was a 1,000-fold reduction in the scale of the problem. These 9, we are now going to be able to go off. There's a hypothesis these 9 might be important. So we are now going to go back and start doing work on discovering better what those proteins do. And this particular hypothesis has been emerging that this abnormal oxidative phosphorylation, or oxidative stress you may have heard more about, is an important part of the disease. And of course, there's literature on that as well.
Essentially, I think what you heard roughly is that we've found a new way to understand cancer. And it's pretty exciting. We found a new way to assess an individual patient who has breast cancer. She may not have been given the proper prognosis. We can see what that is in a better way. We actually have the potential for seeing a new way of treating that person compared to yesterday, and hopefully how to cure that person, potential at least for how to cure that person. We also figured out a little bit about preventative medicine P4, and we see a pathway of working with Lee Hood, who's probably the world expert in this. I think he would say he is, and I think he is. So that we actually can look at health states now in a new way using this new tool, this new system. And you've just heard Larry describe, we can help people who are advanced in their scientific careers find new tracks, new hypotheses, new ways of understanding their work that can actually adjust or change their direction and find more productive solutions. I think it's fair to say all of medicine really, in terms of computing power, until now has been looking at 1 dimension or 2 dimensions, pairwise calculations basically. And I'd like to suggest that until now, if we've been seeing things in 2s, maybe 3s, from now on forward, we're going to see things in a new way. So we're going to see problems differently, and we're also going to see the solutions differently. It's going to look just like this. And this is a picture of breast cancer, the way we see it. So in summary, we have a pattern computer system and it works. And I like to think, yes, it's true, we have been able to create elegant software, really wonderful software, and elegant hardware. Incredible things are going to happen here. And we've integrated them to work. But you know, it's not really what we make. What we make is discoveries. And that's all I've got.